European people like to eat tropical fruit and are delighted about exotic flowers. Therefore, every day, shipments containing plant import products from all around the world reach the subcontinent by different means of transportation. However, such shipments often carry not only the plant products, but also hitchhiking insect pests, such as larvae from harmful fruit flies. Global trade of plant products is therefore seen as a major driver for the spread of insect pests. An example of a harmful insect pest is given by the oriental fruit fly Bactrocera dorsalis. Due to the ability to feed on a wide range of host plants and its high reproduction rate, this species is a worldwide threat for producers of fruit and vegetables and can cause substantial economic losses. Bactrocera dorsalis was recorded for the first time in 1912 in Taiwan, dispersed from there throughout many countries of Southeast Asia and the Pacific region, and is nowadays also known to be present in many African countries. The impacts of fruit fly introduction events are manifold. From an economic point of view, invasive fruit fly pests affect producers of fruits and vegetables directly by reducing their yield, but also indirectly by the consequences of quarantine regulations imposed by the importing countries, such as import bans or expensive monitoring and elimination programs. Pest elimination programs, including insecticide employment, can directly affect the health of farmers and other community members under exposure. Additional health impacts may arise through consumption of chemical residues on treated crops. Beside human health, insecticide applications affect the environment by contamination of water and soil, and may induce toxic effects on native arthropod communities. A cost-effective measure to prevent spread of harmful insect pests along trading networks are inspections of plant import products at points of entry, such as harbors, airports, and border controls. Thereby, plant imports are opened and random subsamples are visually examined. In case of any pest finding, the infested cargoes are rejected or destroyed. In this visualization, we will use pest interception data from Swiss plant import inspections to indicate the extent of fruit fly pest movement by global trade. While inspecting approximately 72,000 Swiss plant imports between 2011 and 2017, a total of 435 shipments were found to be infested with harmful insect pests. Thereby, fruit flies of the family Tephrittidae were the most commonly intercepted taxonomic unit, accounting for 32% of the total interceptions. During the observed time period, a total of 139 shipments from 19 different countries of origin were found to harbor harmful fruit flies. The countries with the highest proportions of fruit fly interceptions were Cameroon, India, Vietnam, Thailand, as well as Sri Lanka. Time series plots show that there is substantial variation in the number of interceptions between the different years. Such fluctuations can be caused by a variety of different factors, such as changes in market demand and supply, pest management efforts by the exporters, as well as new trade policies and regulations. If we take a closer look at the types of infested shipments, we can observe that guava and mango fruits, as well as Java apples and peppers, represent import products which are frequently associated with fruit fly pests. As mentioned earlier, the screening for pest species during the import control is based on visual examination of the plant products. However, the differentiation between harmful and non-harmful organisms by eye can be extremely difficult for non-taxonomists. As a solution, we developed a molecular assay for the rapid identification of the most frequently intercepted fruit flies based on the loop-mediated isothermal DNA amplification technology. The assay is specific for fruit flies of the Bactrocera dorsalis complex, Bactrocera latifrons, as well as Zygodacus cucurbitae. Identification is based on targeted DNA amplification. In case of testing one of the target species, DNA amplification takes place and is measured by the LAMP analysis device. The test result is therefore positive. In case of testing a non-target species, DNA is not amplified and the test result is negative. The test protocol is designed in an easy manner. In a first step, the insect specimen to identify is transferred into an alkaline DNA extraction solution, which is heated up to 95 degrees Celsius for 5 minutes in a heat block. The DNA extract is then quickly mixed on a vortex, followed by a short centrifugation step to remove any liquid from the lid of the tube. Now the DNA extract is transferred into a prefabricated LAMP kit containing all chemicals needed for the LAMP reaction. After a second mixing step on the vortex and a second centrifugation step, the LAMP kit is inserted into the LAMP device and the analysis can be launched. The final result is available within only 1 hour. During the evaluation of the developed LAMP assay, a total of 143 fruit flies from 16 different countries of origin were tested. Of 143 analyses performed, 117 were conducted under laboratory conditions in a reference laboratory, and 26 specimens were analyzed under on-site conditions directly at a Swiss point of entry by plant health inspectors. Thereby, 78 specimens were correctly identified as target species, 64 specimens were correctly identified as non-target species, and 1 specimen was wrongly identified as a non-target instead of a target species. Based on these results, we were able to calculate a test sensitivity of 98.7%, a test specificity of 100%, as well as a test efficiency of 99.3%. As a short summary, in this video we illustrated the unintended movement of fruit fly pests by global trade with its geographic scale and temporal variation, and reported a novel rapid diagnostic tool as a prevention measure. The identification assay reliably differentiates between harmful and non-harmful species within only 1 hour and can be performed by plant health inspectors directly at points of entry.